ahead and get started.
Yeah.
If additional people come, we can certainly continue. But it's my pleasure to introduce Dr. Pradip Singh this morning. And he is here at VCU in Dr. Michael Donnenberg's lab. He got his Ph.D. from the Institute of Microbial Technology in Chandigarh, India, and has been working here at VCU for almost 2 years, year and a half to 2 years. And he's going to talk to us this morning about echo liquid handler and fluorescence polarization as a tool for drug screening.
Thank you. Thank you so much. So, welcome everyone. Today I'm going to talk about the liquid handling systems called eco liquid handler and the fluorescence polarization technique for drug screening. So my talk is divided into 2 parts. First, I will talk about the fluorescence polarization basic principle, how it's work, and then next I will include the eco liquid handling system. So let's begin with the fluorescence polarization. So fluorescence polarization is a technique which measures the change in molecular mobility of a biomolecule. So it can use for several interaction assay, and it provides several advantages over the other techniques, like it's homogeneous. So there is no need of a washing step. You can go ahead directly with a sample, and it's a real-time measurement. So with that you can do the kinetic study. Definitely it's use the fluorophore molecules, so there is no radioactive contamination and there is no radioactive waste. So ratiometric analysis. Ratiometric is something where you compare the ratios. It's compare the intensity of light. So use single wavelength. So in this case, moreover, the concentration of your fluorophore is not going to affect your final result. And then it's a very sensitive, so it's a miniaturizable. You can miniaturize it, and it's miniaturizable. That's why you can do it for the high throughput screening also. So this fluorophore technology technique have wide applications used in this wide area of the research, and like I can't include all those here, but basically like the protein-protein interaction, protein-peptide interaction, receptor ligand studies, DNA protein studies, wherever you feel that there is changes in the molecular size occur, you can use this technique easily. And to understand the fluorescence polarization, we need to understand what is the fluorescence. So fluorescence term itself first coined by Stoke in 1852. And what is the fluorescence? The fluorophore molecule, once it get excited by energy wavelength, a particular energy wavelength, the electron from the fluorophore molecule, it's go to the higher state, and it's remain over there for some time, and then it's try to return back their ground state. By this process, it will release some amount of heat and some amount of the photons, lights, in form of light. So this emitted wavelength are always smaller than the excited one, and there is a time gap that's called the lifetime of the fluorophore. And this lifetime is an intrinsic property of fluorophore molecule, and it's vary with the fluorophore to fluorophore. So this fluorophore have a very beautiful property. If you excite a fluorophore molecule with the polarized plane of light, it will emit back the same plane of the light. The emission will be also the polarized one. So to understand this one, first try to understand what is the polarized light. So in natural, the lights present in the depolarized form, which can understood like that light is electromagnetic wave. So it's have electric and magnetic waves which oscillate to the perpendicular to the direction of its travel movement. I can show here. This is the, if suppose that this light is moving this one, there are several waves which are oscillating perpendicular to the sun, and they are numerous. And to understand the polarization, we don't need to consider the magnetic property. Electric property is enough. So see here, this is the simple representation. The waves, which could be several, but here only 2. So once you are pass through this wave through a polarizer filter, which are the chemical filter, and they have a unique property. They will block all other the oscillating wavelength and only allow to pass one particular plane of the wavelength. So you will get a plane of light. It could be anything like vertical, horizontal, anything. However, you are using your polarizer, this filter. And once you hit with this polarized plane of light to a fluorophore molecule, I am showing here a single molecule which is not okay. So suppose that your molecule is fixed over there in the solution, and by you can assume it's like that, holded by some tweezer. So you will get a same plane of the light in emission, the different wavelength. However, in natural condition, in solution, it is not possible. Your molecule will be moving in the solution due to the Brownian effect, Brownian motion. And as I mentioned that there is some time that's called the lifetime of the fluorophore, which took some time to emit. So once it gets excited and there is a time gap, and by that time it will move to some other orientation, and that's why it will emit a light that will be in some other direction compared to the excitation. That's why in total you are going to get a depolarized light. So this phenomenon was first observed by F. Frigate in 1920, and later on Francis Perrin has did a lot of study on this one and developed several theoretical formula behind this one. And I will come how we measure this one. So Francis Perrin derived a formula to calculate the polarization by using this lifetime of the fluorophore molecule tau and another factor that's called the rotational correlation time of the dye. That is how fast it's moving in the solution. So as I mentioned, the tau is an intrinsic property of fluorophore, so that's remain fixed for a fluorophore. But the rotational correlation it can change, and it is depend on the viscosity of solution, and the V is the volume of molecule. How big is it? That is kind of related with the molecular weight of the molecule. So if bigger, so it will have bigger. And then R is the gas constant, this fixed, and T is the temperature of the solution. So from here you can see you can increase the fluorescence polarization by increasing the molecular weight, and increase also with the solvent viscosity. Like that you are reducing the movement of molecule. I will show in the next slide. And also I found another graph, a graph which is very nice on the website of Thermo Fisher, where they are showing a simulation study with different molecular weight of with the fluorophore attached with a different molecular weight of the molecule, which they are assuming it is a spherical molecule, and from there they have calculated how the fluorescence polarization will affect. So you can see here, if the lifetime is a very small, so there is first the factor is that there will be a high polarization value. However, this small lifetime is not allowing you to measure with the higher change. Kind of you can see when the molecular weight is increasing, this is going to the Plateau conditions. So if you are designing experiment, you need to use a lifetime in which will be related with your molecular weight of the protein. So for example, you can use the 6 nanosecond if you have something which is between somewhere 25 kD to around 10-12 kD. So yeah, and then yes, how you measure this fluorescence polarization? So the instrumentation is quite similar than my previous animation. So here is the source of excitation light, and this is the excitation filter, polarizer filter. It will allow a plane of light, and then your sample, and there is another filter which is called the emission filter. So once you hit this your sample with the polarized light, you are going to get a depolarized light, and with the filter, changing the orientation of filter, you can measure the different intensity of the light. I will come in next. And suppose you are adding here a big molecule with this fluorophore molecule, maybe kind of antibody or something. So what this big molecule will do, it will reduce their Brownian motion in the solution, in the same viscosity solution. So the molecule is rotating slow, so you will get less depolarized light, means you will get more polarized one. So you will have bigger polarization value. So here I am showing instrumentation picture, how it works. So again, this is the light source, this is the polarizer, and you are getting a vertically polarized light here, it's kind of parallel. And then this is your sample source, and this is detector, and before that there is a polarizer. So by rotating this polarizer, you will get two component of the light. if the polarizer allowing to the parallel to this one, so the intensity of the parallel light, emitted light, and then if you rotate the polarizer to the vertical to this one, so intensity of the vertical light. So by using these two parameters, I parallel and I this intensity of the vertical one, you will get fluorescence polarization. So this is a formula where you subtract the two intensities with parallel minus perpendicular and divided by the total intensity. So you can use this formula to get the fluorescence polarization. Similar to the polarization, there is another term called the anisotropy. Both are using the similar raw data. However, there is a slight difference in their definition. For here, the total intensity is the multiplication is the sum of this parallel and perpendicular light, whereas here they consider one more factor, and that's why the total intensity changed by the definition. So it's a parallel plus 2 of the perpendicular multiplied 2 of the perpendicular. So yeah, and then if you assume there is your polarizer is your compound is emitting only polarized light, which is the parallel to the excited source, so you will get a maximum polarization, which is +1, and in other case if it is emitting all the perpendicular to the emission source, so you will get minimum, that is -1. However, in natural condition it is not possible, and you will get somehow in between these, below somewhere. And similar for the anisotropy, so as there, so the formula is different, so you will get +1 for the maximum and -0.5 for the minimum. And these two, as I mentioned, these two are this polarization and fluorescence polarization and fluorescence anisotropy is coming from the same raw data, so it's interchangeable. You can, if you have the polarization value, you can convert into the anisotropy by using the formula like that. Here from polarization you can get the anisotropy, and from anisotropy, IR is the anisotropy, you can get the polarization. And somehow chemical chemists prefer more to use the polarization, and the biophysics guy use anisotropy. However, expert says we should use more the polarization by anisotropy, because this is the definition of total intensity is more close to the realistic value. So yeah. Here I am showing instrumentation for that was the instrumentation for the cuvette model. Here I am showing a little improvement in the instrumentation, how it work in the plate model. So the microreactor plate. So microreactor plate have several sidewise wells. So your sides are not free to get the excitation source or the emitted light. So how it is here? So almost same. Here is a light source, then the polarizer, and then they introduce a mirror called dichroic mirror. And dichroic mirror have a unique property. It is selective for a wavelength, like that. It will allow to pass a particular wavelength. It will like that allow to reflect a particular wavelength, and it will allow to pass particular wavelength. So for, you can choose what wavelength range you need. So here like that it is reflecting the excitation source light, but it is not reflecting the emission, and it is allowed to pass to the. That's why you can use this setting for the microreactor plate setting, and the detector can be either up to this one, or either it can be below to the wells. So then a factor called G factor, which is come into the picture. This the polarized light, the filters and other things do not behave properly with all kind of the light. So we need a correction for this data. So that's correction factor is called G factor. It can be calculated easily by using comparing the data what you are getting from your sample with a known concentration and known polarization value of the sample. So once you get the G factor, you can use that G factor to the similar formula. This is again the perpendicular, parallel and perpendicular, where you multiply the G factor with the perpendicular and you will get the polarization. And similarly, you can use the relative formula for the anisotropy. So This is the basic principle about how this fluorescence polarization. Now I have some experimental example. First, I will include what I am doing, our lab is doing. So our lab work on the type 4 pili system, which is a complex system and present almost all type of the bacteria, and it's considered as the important prevalence factor. So we are not going to focus all other things, but the important thing is that to make a pili, which is a thin appendages, that is made up by the unit protein that is called the pillin protein. So this pillin protein is gets inside in form of prepillin, which have a leader sequence, which is a sequence which give the directions to that. And to get assembled into the pili, this sequence, this leader sequence should be cleaved from there. And if this cleavage is blocked, so there will be no synthesis of the pili. So we are looking for inhibitor which can block this peptidase activity of the this enzyme. So for that, what we did, we use a flash tag. The flash tag is not a fluorophore tag; it's an amino acid sequence, and there are several 2-3 variants of this flash tag, 6 amino acid and 12 amino acid. So what is the flash tag? They have a tetracysteine motif, 2 cysteine motif, 2 cysteine here and 2 cysteine here, and this tetracysteine motif have property to bind with a chemical that's called the flash EDT. It's a complex chemical with arsenic group, and once this chemical is permeable to the membrane also, so it can easily permeate wherever you want in the membrane. And then once this flash tag compound bind with this your flash tag, the complex become fluorophore. So we are using this concept. So we did the engineering to add this flash tag at the end terminal of the leader sequence. So this is the complete, we call prepillin with the pillin and the leader sequence. So this is the cleavage side. So in case when the enzyme is working, so this will get cleaved from the leader sequence, and you will have a flash tag with the leader sequence, which will be comparatively smaller when the enzyme we have an inhibitor and enzyme is not working. So here is no cleavage occur. So you will have a bigger protein molecule with the fluorophore tag. So as I mentioned, the fluorescence polarization measure the change in molecular mobility. So this molecule will have, because this is smaller in size, so it will have different mobility than this one, and it will move faster than this. So by that you can see you will get a different polarization value also. So this is some experimental data where this is indicating where the enzyme is working and it's getting cleaved, and this is a control experiment where I mutated the enzyme, and you can see we are getting a different polarization value which is higher than this one. So we are using this one to screen for inhibitor, and we are doing screening for the different chemical library. So I'm not showing all those data. And second example, I'm showing here a kit which is made for the prostaglandin desynthase inhibitor. This is provided by some Steinman Chemicals laboratory, and this have a principle. What they have did, they have developed a probe which have a tendency to bind fluorophore probe, which is a small in size and which have a tendency to bind with the active site of the enzyme. And once so if you are screening the inhibitor, so if you have inhibitor and have a tendency to bind with this active site, it will remove the probe in solution. So here in actual condition, in the initial condition, you will have a probe with your enzyme, so it's a bigger size. Whereas after if you will get a inhibitor, it will replace that thing, and you will get a smaller fluorophore molecule. So you can you will see a decrease in the fluorescence polarization. So yeah, and they are showing some control study where they have use this kit, and these are the known inhibitors, so it's fine. And then another example, that is the Transducer ADP2 assay kit, and they have this kit is developed to screen the ATPase activity of enzymes. So usually the ATPase kinases are once they react with the substrate, so they phosphorylate us, they do something, but the end product will be ADP also over there. So what they did, they use antibody which is specific for this ADP, and they added a modified ADP with the fluorophore tag. And once you have the reaction is goes in the positive directions, you will have more ADP in your solution, and then this ADP is going to replace this fluorophore ADP, and again you are going to see a change in the fluorescence polarization in presence of this enzyme. So on the basis of this concept, there are several other kits. So yeah, so these are the few examples, and similar to this, there are several lot of examples, and people are using very in the diverse area of application. This fluorescence polarization technique is the kind of very established. Oh, so yeah, another. Once you go for the high throughput screening, a factor called J factor come into the picture. That is a factor which is developed by Singh in 1999, and this factor gives you a big idea. Your screening, if you want to use this this technique for high throughput screening, so your your model is going to work for high throughput or not. So for that, they use standard deviation of your positive control and negative control. So these are the standard deviation, and then this is divided by the average subtract of the average value of your positive and negative control. And if your G factor come above the 0.5, you are very good for the go for screening. But if G factor is lesser than that, then you need to rethink and design another thing. So this is a really good thing to know is your experiment is going, if you are going to screen a large number of library and something, so you should calculate your J factor to validate your model is working or not, will work or not. So this is all about the fluorescence polarization, and here is some reading material if you guys like. So I like this book. This is from Joseph L. Lopez from University of Maryland, Baltimore, and this is really very good for the spectroscopy. They explain from A to Z, and there are a few reviews. So yeah, this is from the fluorescence polarization. If you have any question, then I can go further for the liquid handling system. Okay, so go for the liquid handling system. So yeah, as I mentioned, for G factor, there is a standard deviation. So we can assume for high throughput screening, liquid handling, the handling of the liquid is should be very accurate to reduce the G factor also, and also for other factor also. So in tradition, the liquid handling system use tip, capillary, or pin, something like that, and these tip, capillary, or pin have a different interaction property with the solution if the solution have different viscosity. Like that you can see here in the picture. The tip, if you is example, if you have a different solution in your tube, so these same tips are going to behave differently with different viscosity of the solution, and that's give really large amount of error once you go for the smaller volume and high throughput screening. So here I am going to talk about a liquid handling system which is a totally unique property than the all traditional liquid handling system. That's called the eco-liquid handling system, and it's used the acoustic energy to transfer the liquid, and it do not use any tip, pin or anything like that. And this is really a smart instrument, and it's determine the fluid composition, all those things. And I will talk a little more in detail here. So this instrument gives a lot of flexibility as it's used the acoustic energy, and they have designed in a such a way. So there is a lot of flexibility compared to the traditional system, like that you can transfer fluid from any well to any well. There is no limitation like that you are going to row to row, row to column kind of that thing, and from a single well to multiple well, a lot of things you can. And then again, it's not using the tips, capillary, anything, so there is no further washing step required, and you can use empty well of filled plate, and this is very precise, fast. So, and also there is no chance of the cross contamination. So how it work? Okay, first there is a comparison. I think this is quite old, but I tried to find something newer, but I didn't find any new comparison. But this is okay to understand with the how it's good. Like that here they are trying to show with compare with the other liquid handling technology which work in the low volume, and you can see this is the data for eco and this is CV. So for maximum instrument the error rate percent CV is the error rate, which is higher in the lower volume range, whereas eco shows really good, very less error. So how it work? Eco use the acoustic energy to transfer the liquid from your source plate to the destination plate. So in practical, it generates the radio frequency wave which transfer to the transducer, and this transducer convert this wave into the acoustic pulses, and then these pulses hit your source plate, and then with the proper enough energy, that's release a drop from your source plate, and this drop will fly in the air, and here will be your destination plate, and that will get attached with your destination plate due to the surface tension. And system is so smart like that it will automatically calculate the energy, how much energy is required according to the volume of your sample and viscosity and all other properties. So it's required really less calibration for the designing your experiment. So yeah, here is a pictorial way how it works like that. This is the transducer, and it can move beneath your source plate, and it can go here to there anywhere. And then this is also movable, and then like that here it will automatically calculate what is the volume over there, and according to that it will generate the energy to transfer the liquid. And this do very fast. The droplet generation is really fast, like 200 to 500 droplets per second, and the single droplet have a fixed volume of 2.5 nanoliters, so it's very accurate also. So here I put a video picture how the instrument do the inversion of your plate, like that. Here I'm showing the plate is inverted, so you have to just put your plate on this handle, and then it will rotate back and take it inside. So it's quite okay. And with this system, at this plate, in the plate you can use several other kind of plate also. So okay, here so you can work like the 96 well plate, 384 well plate, 50, 134, and I think above that there is a 34 and 56 well plate also you can use. And in that, in place of the plate, you can use other things like slides and other things which can be attached with that handler system. So it's good. And absolutely there is a surface tension work. So if you have some amount in your plate, your source destination plate of liquid, it is not going to fall down. You can check it by just inverting that. If it's checking, so you may need to reduce that one. So that's good. However, for source plate there is a limited plate because they need a constant thickness of the bottom. So this plate is provided by themselves. So it's a 384 version plate and 1536 well plate. So according to use, you can choose the plate. And there is data which showing this system will work with the several type of liquid, like crystallographer use different kind of liquid, TMS, so buffer, surfactant, glycerol. So wide range of liquid it will work, and so maybe very little amount of the calibration required if you need. That is also one-time calibration. Okay, so for the handling, there are several software to make your work easy. So this instrument is present in the VCO actually. So there are several software. So right now we have the eco plate reformat and this dose response. Maybe we are going to get sooner this cherry cake one. So they make easy. So here I am going to present how this eco plate format work and how this system actually works. So as I mentioned, you can transfer well to any well, like that one well to any well, or you can do anything. So here you can see the color code is showing. So this is my source plate, and from here I am transferring in the all the column, and here you can see this is the row. So you can do the transpose also like that from row and transposing to the column, and column to row, and from one belt to all plate over plate. So it's a really very flexible instrument and give you a lot of freedom to work with your library compound. So this is again the pictorial presentation like that. Here from one to from different sample you can transfer to the one belt. So you can design as whatever you want to experiment to do with yourself. And in further application, like if you want to transfer your 84 wells, some sample from 96 well plate samples, so and you want to distribute uniformly at different ways, so you can do this one, and you can do the not reverse from 96 well, but from 1534 yeah, from this one to the 1500 something. So from any well, any plate to any plate. And this is a presentation for dose response. So like that here they are showing by using the echo. This is the traditional way how we use the like that. If we got some heat compound, so how we work further for the dose response. So we'll take out into the one plate, and from here we will make the serial dilution. That is a several step of the handling, and then from the serial dilution we'll transfer to the assay plate. But if you are using the echo liquid handling, so from source plate you can directly put the assay plate. And you need maybe one intermediate plate where you can put a different concentration to make 12 point dose response. So it's reduced a lot of effort like that. Here you have to do several steps of the pipetting, and which lead further contamination, cross contamination. So it's quite really fast. And this is the graph they are showing the thing. And at the end. This instrument is smart enough, as mentioned, so it will calculate, it will give you the log value of your transfer. Like that here I just want to show there are some error here. It's showing there is no volume transfer. I supposed to transfer 40 microliter, but actual volume transfer was 0. So why? So the volume is higher than the expected volume. So it's work in the source plate, the sample volume should be in a particular range. Like for this plate, what I'm using, it should be between 20 to 50 to 65 microliter something. So it is above than that one. So it's given transfer from there, and it will also give the composition of fluid. Like that here is saying 74% of the DMS. So it's really smart. And this instrument, a lot of people using, and a lot of applications from this clip from their website, and they have shown the application of this instrument in the several areas like the drug discovery, genomic, and cancer research, and all those things. And you can find several good presentations like that. I like their crystallography presentation over there, and they are showing how this is very useful to reduce the size of their experiment from like that several microliter. So like that saving their sample of the cast sample, that thing. So yeah, and this instrument is present in our VCU facility. So yeah, this is here on the echo. This one, and I'm showing this the Clariostar, which is the fluorescence polarizer spectrophotometer. And yeah, that's all. So yeah, this is all about the presentation, and I would like to thank Dr. Aranme, the director of the IST facility, and Heather for helping me to understand this instrument, and Dr. Srinivas here, the facility manager, core facility manager here, and my PI, Dr. Dannenberg, and of course all of you and the guys, the library staff, to arranging this talk. Thank you, Stacy and Smith. Thank you very much.
So I thought that was a wonderful talk. And one thing I was just, the ideas being formed is, were these transfers sterile? Like can you do this in like a sterile context, like if it was with like cell lines and
that's really thing. So I think There is no contact. First of all, you need to understand there is no contact occurs. So if your environment where the instrument is kept is sterile kind of something, so the transfer will be sterile.
Yeah.
But I like that again. If it is in common facility, so you need to think. You can maybe answer. Like we have the representative from the lab side
Sure,
also. So
sure.
would you like to answer the question?
I'm
Yeah,
Mariana
so-
Mott. I'm the account manager for LabSite, and that was an awesome presentation.
Thank you.
The Echo uses sound energy, so there's no tips, no touch, no contact whatsoever. Your inverted destination plate is very close to your source plate, and it shoots directly up. So there's no cross-contamination or anything. We've actually done experiments in, using qPCR and 1536 well plates, and where we put a positive sample surrounded by negative samples, and no cross-contamination was seen ever. With regards to environmental controlled, it's not, but the Echo is so fast. We have people doing, dispensing small compounds or a variety of reagents onto cells. And actually, the Echo is so gentle in dispensing, we actually have people shooting live viable cells.
Very
So, yeah, it's a really cool instrument.
What happened to your Z factor when you switched from pipettes to Echo? Remember like numbers?
Z effect. Yeah, it's improved. So, like that, when I was working with a multi-channel pipette and 96 well plates, so that time it was really tough to get a Z factor constant. It's come around 0.5, but not always 0.5. But when I shifted to over there, the reproducibility increased. So at least for a particular set of experiment, I'm getting a better value than the handling of that one. So manual handling. So I'll say, Michael, like that. Once you do the experiment, so as that is a live cell, so it's very plate to plate. But maximum time I'm getting good one.
Better than 0.5?
Yes, 0.5, 0.6 something. So in a way like that, it reduces a lot of standard deviation. It's reduced significantly until this I'm not doing something with the cells wrong. There's some defect.
Okay, so the sound frequencies that the instrument used to transfer the solutions, does it depend on the solution itself, or like how does it determine what frequency to use?
Yeah, as per I understood how that works, so first it will send a sound signal to calculate the viscosity and the volume of sample, and once it will calculate back, and then once you put the plate, it will do the scanning for all the wells, maybe at that recall for all those things, and then according to the viscosity and the volume, it will decide how much energy it's need to send there to generate a 2.5 nanometer. I'm right?
I could, the Echo, as you pointed out, is a very smart instrument in that it uses what we call dynamic fluid analysis. It actually, before it shoots or does anything, it'll survey the source plate with your reagents, and it sends out 3 pings of sound. It hits the bottom of your source plate, the bottom of the well, and the meniscus. And through the various algorithms that we have built in to the Echo, the calibrations, it can determine, because it also listens to the sound, the echo from the sound, and it can determine the volume as well as the various properties of your fluid, like the viscosity, percent glycerol, the percent DMSO, and so that when it goes to shoot, it knows, so it produces enough sound energy to create the mound to dispense your drop repeatedly. And it does this very fast, 200 to 500 drops per second, from any well to any well. So what would take you manually or using another liquid handler can take like seconds or minutes to set up a plate. So it's really fast.
The only consideration is that you have to be careful about organic solvents and the aqueous solvents. So you can't do both at a time. So you can do aqueous first and DMSO later, something like that.
Mm-hmm.
So I have to specify the algorithm also what solvent system you have, to at least to be brief. Not exactly how much amount of DMSO, but at least you should say DMSO or water, something like that.
Mm-hmm.
Yeah, question, if you have like two, you first put the DMSO and then put another solution, can they mix well? It's not like, you use pipette to mix the solution.
It can mix probably. When you do it separately and then you can ask the instrument to mix it, maybe, yeah. But we haven't tried that part.
So I have a question. Is there something like the coming with the tubes as a source plate, or still we need to use that 384 well plate as a source plate? So what other option they are coming with further?
So the reason why we require our users to use an echo qualified source plate is because the dynamic fluid analysis, remember I told you the sound it listens and puts out the pings of sound on the bottom of the source plate and then on the bottom of the well. So that depth of thickness of your plate really needs to be consistent so that we're continually, and accurately shooting the 2.5 drops. So that's why we require a source plate. Currently we have 384, 1536, and then we have another Echo version. It's called our Echo 525 that shoots 25 nanometer drops, so bigger drops, and for aqueous solutions only. And that has an additional source plate, which we call our 6 reservoir well, because that's used more in the genomics market, where setting up PCR, sequencing, doing DNA assembly, CRISPR, transfection, you name it. We're just miniaturizing everything. So, does that answer your question? We are looking to create a 96 well source plate. It's not out there yet. And it'll probably be more driven towards our Echo 525, just because in those assays, you're using a lot more reagents. I mean, you're going from 50 microliters down to, we can go down to 2 or 5 microliters. So,
All right, thank you so much.